Here we go. So, as I said, so I think the bookstore was trying to, again, get these so they were on black. Why, I don't know. Because apparently, if you're going to use the Connect, and it's not mandatory, and we went over this before with regard to the cost or whether you have to have it, and it may actually be accessible now. You can see if, because it's my understanding for the first 2 weeks, you may not necessarily need an access code, but yeah, hey, use the link and go from there if you want to see what it's like. Fine with me. Okay, and so that's the information that's there, and if I get more updates, it's really my one of my colleagues, Dr. Maurer, who some of you might have for AP2, is much better at doing this than I am. Then again, anyone is. So much for that introductory stuff. Let's get where we were. Okay. And so where we were was this with historical figures, and where I left off, we had talked a little bit about Jenner with vaccines and cowpox. We had talked a little bit about Semmelweis and really sort of landmark stuff about puerperal sepsis. And honestly, that was when really the transitions, people were really understanding that not only was there stuff in the air, but there was stuff in soil, there was stuff in dead tissue and dirt, grease, grime, whatever you want to call it, and was able to, begin to get this. And then we got to Pasteur, and because of Pasteur's credibility, this is really the start of what modern physicians called germ theory. And germ theory is this: that there were these particles that you could not see that you and I ubiquitously call germs, even though it's sort of a global term. And we use that here. We'll talk about germicides, whether you're using bleach or you're using alcohol wipes or you're using some kind of disinfectant. And we'll go into all the nuances about that as well. But he really was a landmark physician. And what he did, and you saw I wrote it on the board last time for those that may not have reviewed it there, is because of the wine industry, he had looked at wine that was spoiling and found little particles that were there because they were coming from the yeast that were there, and there were probably some bacteria. And what he did was he realized that if I boiled the wine, it wasn't going to get anywhere. It was going to lose what made wine so desirable. So to avoid the spoilage, he was able to, and I'll write down the numbers again, and we go over them and when we do our sort of our unit on methods of control with sterilization and things like that. And what he came upon was a method, and we'll see how this is not necessarily used as much today as it was then, though. To be sure, if you're buying a vintage bottle of wine in France or from a French vintner, let's say at the state store, they're doing this method. 30 minutes at roughly 62.5, so 62 or 63 degrees Celsius, which is about 140 degrees Fahrenheit. And that, by holding the temperature steadily there, he was able to maintain the quality of the wine and effectively stop the spoilage. So that immediately made him credible. So what did he do when when this whole idea, which was everyone was talking about, was germ theory? Is there a vital force in the air, or are these little particles we can't see? And so that's really where the story left off. So I'll go back. This is the Nestor PowerPoint, and because it's some of those. This was when I was telling you about von Leeuwenhoek, who was the fabric merchant. He called them animalcules. And and I know you're starting lab, when you first start to look at things in lab, you're going to see under the microscope little circles and little rods and some elongated and some spiral figures. And it's exactly what he saw, magnifying things about 300, okay, 300 times. The other, whose name I couldn't recall, is Robert Hooke. And he basically saw what effectively was bread mold, which is everywhere, and we all get it, and we've all seen it on bread that's lingered too long. That's penicillium. They have a very distinctive way of reproducing. They're called spores, and they look like little mushroom heads. Okay, and so he was able. That's where he came with that. So and you'll see that. And then this is what they used to do in some science classes. Was actually, this was the original design for the von Leeuwenhoek single lens microscope. And then we start talking about foundationally how long these things have been around. And by utilizing carbon dating techniques and fossilized remain, they were able to determine, and collectively we use the term microbes, because a virus and a bacteria are certainly not the same. As I commented on before, viruses by definition are non-living because they're not made out of cells. So with the term we use is microbial. And nowadays you'll hear antimicrobials because antibiotics are a subset of that. Anti-infectives, antivirals, because they're a subset of antimicrobials. So terminology is important. And always from a test taking perspective, that's important. And that's the kind of little nuances that you see in exams. And for those, and most of you have had me before, you know how annoying that can be. It's all right. You know how good I feel whenever I go, I hate your exams. Oh, thank you. I didn't get you anything. So so the ancestral bacteria based on fossilized remain, they dated, whatever you make of it, 3.5 billion years ago. I wasn't there, despite what a lot of people think. Okay. And we'll go into the importance of microbes and things like that. This was the spontaneous generation nugget, and it took a lot of time to refute it. This is the one I talked about before with Reddy and the rotting meat and the worms or maggots that he saw. And look at the time, 1668. And again, most of us think there were 3 jars, not 2. He used fine mesh gauze, which is effectively cheesecloth. He was in Italy. They love to make cheese, formaggio. Okay. Hey, that's what I'm talking about. And covered with 1 jar with gauze, preventing the flies, no eggs, no worms. And again, there was some, it was the one they couldn't duplicate it because they didn't necessarily have the same cheesecloth, and that's really the concept of validity. And 2, the naysayers, that cloth, that gauze, that drove away the evil force in the air, whatever. And then they used a lot of broths. That's an important little nugget that's coming up here with Pasteur. So this fellow Needham demonstrated boiled broth still produce microorganisms. Well, the little uptake from that, as you're going to see very shortly, is boiling is not the equivalent of sterilization. And that's something you need to be very, very familiar with, and very, very soon. There are some things that can be sterilized by boiling, but not everything. And so what they used a lot of, and this is where it sort of drove into Pasteur's work, they used a lot. What an infusion is. A cup of tea is an infusion. A cup of coffee is an infusion. All you're doing is you're using water to extract, whether it's flavor or particles or material, from typically plant or plant-related stuff. What they used, so the broths that you're going to see that Pasteur used were similar to the wine. They involved yeasts and sugars. That's where his problem was with regard to his experimentation. What was used a great deal, particularly by Needham and others, as you can see, within 25 or 27 years, this father Spallanzani contradicted results. Boiled broths longer, sealed the flasks, they remained sterile as long as the seal was intact. The question was they were not uniform, and that's the idea in science of validation. What Needham was using were called hay infusions. What they used to do is they would pick the hay that was left on the ground after they made hay and throw that in a vat with any other plant material. Well, what's on there? Dirt. Okay, aha. What's in the dirt that's not remaining sterilized? So, argue the heating destroyed the vital force. Well, nobody cares. So, what happened is Pasteur worked with the glass blowers, and he did some studies. He started to look. He would basically filter the air through cotton plugs, take little slices of cotton, and look under the microscope. And he saw organisms that were similar to what he saw in the wine. So that was his aha moment. Okay, so they found them in a variety of these broths and wines. So he reasoned they were coming up into the plugs. But plugs in sterilized broth even still gave rise to microorganisms. So he realized that there were these tiny particles were likely to cause the problem. And so he had to construct something that effectively allowed exposure to the air, but not directly so. So he made this sort of gooseneck or swan neck flask, whatever you want to call it. And so he boiled infusions. okay, remaining sterile despite being, because they were open to the air, the neck piece. And what he reasoned was as he boiled them, that the particles that contained the problems became trapped in the bend. And as long as they stayed upright or unbroken, they remained sterile. But as soon as they were tipped or broken, immediately, and this is what you're going to see in lab, broth that is sterile is clear. It's a solution. No matter what's dissolved in there, as long as it's not large enough particles to deflect the light, it's clear. That's what means it makes, when you have a tube of sterile broth or sterile water, if you hold it up to the light, it's clear. If it's cloudy at all, something's going on in there. That means it's contaminated. So if you're doing an experiment in the lab, you don't use the broth. By the same token, if you see something you shouldn't in a hospital sense, and something you're taking an injection out of, and you're like, whoa, is it supposed to be here or not? Some stuff has stuff that's dissolved in it. Various medications you have to shake. They become what's called in chemistry a slurry. And we use them a lot. So you have to know what you're injecting. It's not always going to be clear. That's there. And so, and very, very shortly, the problem was right here. People could not duplicate what Pasteur did because they didn't all. He used a broth that was made of sugars and yeast, very similar to what he studied with his pasteurization experiment. So when he boiled them, they were fine. But others were using this hay infusion. And so they began to study this a little bit more. And as they did so Tyndall conflicting data and showed Pasteur his reasoning was correct, his methods were not. Sterilizing broths different times, 5 minutes for some, sometimes hours didn't make a difference. So the hay infusions with the particles from the ground, the labs that could not reproduce Pasteur's result used broths made from hay. And so at that point, this German botanist named Kohn discovered, when he did microscopic analysis of particles in the hay, something called endospores. Now, endospores, and what I alluded to before with penicillium spores, not the same. Penicillium is a fungus, that's a eukaryotic cell, it's different. Endospores that come from bacteria are really, as you will come to find, the definition of sterilization. If you kill the endospores, it's dead. Now that you're going to see a nuance to that down the road with something called prions that are not included. So whenever you see contents are guaranteed sterile, like on a Band-Aid, unless this package is open, it could be teeming with prions, not that it is, but they're not included at this point in time since we do not know scientifically what the definition for sterilization of a prion is. You'll see this down the road. So as you can see, Pasteur used broths made with sugar or yeast extract. So you had to reproduce. That's what validation is. So what are these magical particles called endospores? Yet every time someone goes into a hospital emergency room with a cut or a wound, literally the first thing that they ask them, maybe how'd you get the cut? Are you, right, do the vital signs, but immediately in that initial interview, they ask them something that is addressing the concept of endospores. What's that? What are they asking? You're in the hospital, they got to be asking you something. When I go to the hospital, I know what they ask me. Here's the first thing that they ask me: health insurance. Because they want to get paid. Thank you, RMU. I'm sorry, I have that other card in my wallet, the one that I don't show anybody. It's coming out. Oh, it's in here somewhere. I hide it. My Medicare. That was on Monday. It was my wife's birthday. She just got hers. She's refusing to acknowledge. But I digress. So what do they ask you after they ask about your insurance information? Do they ask you about something that's up to date? What do they ask you about? Yes, ma'am. Allergies. Good point, but not the answer I was driving at. But that is an excellent point. Absolutely. Nick. Tetanus shot. Correct. A tetanus shot. They want to know if you had a tetanus. Why? Because any wound potentially could be a tetanus-prone wound. Any wound. Name of that organism, Clostridium tetanus. We use binomial nomenclature. Aha! What's interesting about tetanus, it's an anaerobic bacteria. In the air, it doesn't thrive. But in a wound with a lot of dead tissue and without adequate blood supply and oxygen, it can begin to develop and creates a toxin that is deadly, absolutely deadly. So it doesn't always have to be what you and I would think it was a wound. More on that down the road we get toward the end when we start talking about wound infections. So that's the big player. You're constantly, by and large, wounds that come into the emergency room to be sutured are not tetanus-prone wounds. Those are usually pretty nasty wounds with a lot of dirt and stuff and irregular edges. A typical kitchen knife wound needs 2 or 3 stitches, not a tetanus-prone wound. Why are they asking you about that? Because maybe that time that you're out somewhere and get injured and they can't get to you fast enough. So they want to make sure that you're tetanus current. And you get not 1 immunization, you get 3. It's called DPT, diphtheria, pertussis, and tetanus. So when you're giving a tetanus shot, you're actually getting 3. More on that when we get into those disease entities. So that's an endospore. We're always worried about it. Why? Because effectively they're indestructible. They're always with us. On this carpet, endospores. Bottom of your shoes, endospores. They're there. You want to have some fun with it? Grow some in the lab if you can, and take a look. Okay. I had a student who was doing home brew with honey, which is basically mead, years ago. I mean, because there was some funky stuff growing in it, he brought it in, we cultivated it, and what was that endospore that grew in honey? It's a different one that you've heard of. It's the one with consumables. There's really only one kind of food poisoning that's immediately deadly. It goes by this organism, you're going to see the trend. Clostridium botulinum. Very famous disease. And food poisoning, if it's not addressed shortly with an antitoxin, you have to actually give the person the antibody that destroys the toxin, not the infection. They get a terrible disease called botulism. There have not been a whole lot of cases. You don't see a lot of them. There was a famous one out of Newark, New Jersey. Right by Newark Airport, there was a company that made cold potato soup, which is known in French cooking as cheese. And apparently they didn't do the right thing. There's a way of canning, you have to be very careful, particularly with non-acidic things. And they had about a dozen people die from that. He was sitting right out there, so right by the old Newark Airport. Clostridium botulinum, yet we take this extraordinarily deadly toxin in microscopic amounts and purify it. What do we make out of it? Botox! So we use therapeutically for a lot of things. So sometimes they don't do anything with tetanus toxin, but botulism they do. In war, not so much modern war, you don't see a lot of it. There's another Clostridium. All these are endosperms. All these are anaerobic adaptation. Okay? And there's a variety of ones that are sort of cousins of this. So if you're like me, not many people are these days, I'm a World War II. One of my favorite places in the world is in New Orleans, the World War II. I could just go there. And see the world, just immediate. So much jungle on it. Fried dough. Clostridium perfringens. In World War I and World War II, they ended up with a lot of amputations. The reason was this, because they didn't have helicopters. If someone was injured badly, they had a couple of ponchos, and they put them on a couple of rifles, and you stretchered the guy miles to try to get help. And if you got there soon enough before gangrene set in, and this is what it caused, gangrene. There's more than one type of gangrene. But this particular gangrene, really the only treatment they had once it set in was interestingly, one of the agents we're going to look at in antibiotics, okay, comes from one of the original early antibacterial substances called sulfanilamide. It was in wound packs. Soldiers then had a green powder they would sprinkle on the wounds. It was played a major role in why the Allied forces defeated the Axis forces. More on that later. So there's another one. Why? Endospores, dead tissue, anaerobic environment. And the one most of you are going to see is this stuff. Clostridium. Are you ready? Difficile. Difficile because it's so difficult to get rid of. What's the name for it? C. diff. Because in micro, all you have to do, you use the first initial. So you could write C. difficile, and what we've always abbreviated to is C. diff. And let me tell you, ladies and gentlemen, if you're in a hospital, you can make that diagnosis from outside the room. Why is that? It's normally in our gut. When you take certain antibiotics to kill the other bacteria that suppress it, in your gut, deep inside the colon, is an anaerobic environment. It can flourish inside. I unfortunately had that. And it sort of indirectly came from my wife at the time, who was running a nursing home. She was the nursing supervisor at BSN, RSN, RN, that type of stuff, at a nursing home in Wilkinsburg. It's not there anymore, but it's a big one. It was the old Wilkins house. That's just a start. Now, interestingly, that's why Pasteur had the right idea, but didn't have the right experimental stuff. And so he went on to be very, very famous because his idea was treatment. Without knowing about, he coined the term virus. Without knowing that it was a virus, he developed, the first way of treating rabies by extracting, effectively serum, which had antibodies in somebody's system, doing something to purify them and injecting them into other people. So he really developed the first actual treatment because rabies was totally deadly at that point, and it still is if it's untreated. So he did a lot of stuff, particularly and developed the Pasteur Institute in Paris, and that was and extraordinarily famous. And if you go back and look at all the things that he did, it's quite remarkable. At the same time, not necessarily the same time, a little later, he had a rival, but a friendly rivalry where they each made discoveries. And that gentleman's name was Koch. And he was in Germany. Interestingly, he won a Nobel Prize for his work. Pasteur did not, because Pasteur died prior to the start. He certainly would have won one, but he died before the Nobel Prizes were started. And the rule is you must be alive at the time it's awarded to you. Not necessarily presented to you, but you have to. And that's happened where someone has died shortly after being notified. Koch received that award, might have even had 2 times. And Koch was where Pasteur was the clinical guy helping people. Koch was the lab geek. His whole idea was identifying germs. So he was what you're going to use in the lab, and you probably have used it before in other schools, is agar. It's sort of like this gel-like stuff that you grow bacteria on. It was his lab that developed all those cultivating techniques. The streaking of plates, he did that. Most of the bacteria that cause problems, he identified. Things like tuberculosis, staphylococcus, E. coli. He was the first one to grow them and identify them. All of those. The one endospore that he was most famous for addressing was this. Now, this is an endospore that's all over the place, which is not anaerobic, meaning that it can grow anywhere. You use a cousin of it in the lab called Bacillus subtilis, or what we call B sub. And Bacillus subtilis is very, because you get to study them and they're easy to grow. This one is called Bacillus anthracis. And it causes a very famous disease, anthrax. And why Koch was so well known was that he identified anthrax and found out that that was in sores that were killing sheep and cattle and had a tremendous impact on the European economy. And he developed at the time, we don't really use as much today, but there's a foundation, like the chain of evidence, that supports how something causes a disease. They're called Koch's postulates. And we'll look at them subsequently. Basically, what he had a disease, had an animal was suffering with anthrax. He pulled fluid out of the sores. He grew in the lab the bacteria on a culture plate. Then he extracted from the little areas of growth okay, what are called colonies. He took those and injected that material into a healthy animal that was susceptible, watched them to get the disease, unfortunately die, and he could do the whole thing over again. That effectively is a synopsis of his postulates. Very, very interesting. So those are really foundation, and there's more. There's a whole timeline on this PowerPoint of all these amazing microbiological discoveries. The way that when they discovered basically the way that we can do DNA analysis and genetic engineering and gene therapy. And there's a difference. Genetic engineering is we're taking these enzymes that you'll learn about called restriction enzymes out of bacteria that chop up DNA. And then what we can do that is basically develop ways to make the sequences, which we can do in the lab, and put them, and they're used all the time. All the time. Okay. The first genetically made material, insulin, 1977. Insulin, which they knew for a long time was the way to treat diabetes. Initially, insulin was made from pork. Okay, so they would take, and it goes in the pancreas, which you'll get all about this in AP2, many of you. It grows in the pancreas. They would grind up the pancreas and extract the insulin, which is a protein, and whatever they did to it, they'd inject it. And there was a lot of people were allergic, and a lot of people, for religious reasons, did not want to have a pork product that were there. My grandfather was a diabetic who didn't want. I don't think his refusal was so much because he was Jewish, but more because he didn't want to take the shot. So he died relatively young, like at age 70. Not young for that time, but I have a very faint recollection of him back in 1955. And then when I started to study about it, they were making the insulin from cows. It was bovine insulin, and that was the insulin that was used extensively. It's called NPH, it was named for three doctors that had put it together, and it was a lot more accessible. There was a lot of cows that were being slaughtered. People at the time thought they were healthier, and there was less objections to using them. All well and good. Okay, and then about 1977 was the development of Humulin, human insulin, that was made in the laboratory, directly mirroring the sequence of our insulin. So it was just the ideas of having allergic responses to it, or people objecting to it. And really, that's the kind of insulins that we use today. It's not like they're killing people and grinding up their pancreas to do that. They're making them in the laboratory. I'm just saying, as far as I know. So that was the first. Oxytocin, known as Pitocin, which is a very simple peptide compound that has about 9 amino acids that you'll learn about again in the endocrine, and something called vasopressin, which are made by the posterior pituitary. And the Pitocin is the genetic thing that's used to induce labor, because normally endogenously during labor, that's produced. It has other things that it does. So those are just some of the examples. Hepatitis B immunizations most people have had from that. So that's the way human growth hormone that people may have used that we can use therapeutically. They're relatively simple proteins to make. That's just some of the idea that's there. And I hate it when they do these golden age things. Okay, great. Basically, you'll see that historically in science, from the time roughly of Pasteur and then moving on, we saw a lot of changes that were there up until maybe the start of World War One. Then during world wars or giant conflicts like that, the scientists tend to go into do things for the war effort, mostly for bad rather than for good. Then and then in between the wars, there was a certain amount of scientific work that was being done. Interesting, most of that was done vis-a-vis atomic and nuclear stuff. Then that was put away, and then scientists started working on nuclear weapons. We know it had eventually spawned the bomb that was used at Hiroshima and Nagasaki. And then they resumed things like Watson and Crick when they knew about DNA, and that was being studied before World War II. Then the timeline was that became, and then DNA became the thing that was from Watson and Crick, and they were competing with another two-time Nobel laureate named Linus Pauling, who was out at Caltech. So just, I mean, just little things. And there's the timeline thing. So if you're interested in that, take a look. Let me go back to these historical figures as I jump around here. Okay, so there are all those neat bacteria that are there. Here's Koch, lab-oriented, pure cultures, his postulates. The one I didn't bring about was this gentleman named Lister. Lister was inspired by Pasteur, and he is the father of what's called modern aseptic technique. What you will, all of you going into any healthcare related field, is all about aseptic technique. It's just, we're much more sensitive to it now because in the COVID era, we learned about that and began to utilize it more extensively. Okay, all of a sudden we became very aware of it, but in medicine they've been aware of it for ages. Lister made an observation back around in that like 1860s, 1870s area, and he saw that when they were operating on people, that basically they'd wipe off the instruments, not necessarily clean or wash them. They didn't put anything on the skin. They didn't really do a whole lot of hand care, and they would operate, and there was a tremendous amount of infections. And so he began to work with dilutions of a very common substance called phenol. Phenol in the time was known as carbolic acid. It was a cleaning solution. It's very, very caustic, but when you dilute it, okay, it has interesting properties. If you dilute it sufficiently, it can even be a local anesthetic. And if you dilute it even more, it's a very useful agent that can act as both an antiseptic and a disinfectant. Down the road, you'll get the distinction between the two. Antiseptics you use on skin, disinfectants you can't. You make it brief. And what happened was he had these phenol solutions that he would cleanse the instruments and wipe off people, okay, with that, and have people wash their hands in it tremendously. And that was the start of what's called aseptic technique. It's a different, the word sterile technique is somewhat of a misnomer. We can sterilize things, but once they're open, they immediately start to become unsterilized. So you'll hear, do not tell your nursing instructor. When they say, we're going to do sterile dressing changes at bedside, Dr. Stein said they're not sterile. Don't do that. It's enough that you'll know and I know. Okay. You just look at it and go, yes, okay. Yeah, let's do that. I know. I'm evil. And there's the anthrax thing. Okay, moving back on. Let's get back to the other PowerPoint. I don't mind jumping around like this at first because it's good to do the introductory stuff because I'm trying to follow this. And remember, this is the one we looked at from them, and we talked about analyzing them and indirect means and all that. And then they ask you these questions. And, okay, so this is going back again to that billion years ago. And the distinction, and this is really the hard part, and I spend a whole lecture on this, showing you the nuances and the distinctions between eukaryotic cells, our cells. Eukaryotic means means to a certain extent you are true, and the carrion is like the core or the center, or it's and so it's almost like the kernel in the corn, if you want to think of it that way. And so it's gone on to mean true nucleus, but effectively eukaryotic cells, and we learned about this all in A&P, membrane-bound organelles, of which the nucleus is easily the most visible. Where bacteria, single by definition, no true nucleus, and they're called prokaryotes. So pro for pre or before, and bacteria and its cousins, archaea, which are the oldest, okay, are pre-nuclear. They do not have membrane-bound organelles. They do have DNA, but it's not within a membrane-bound enclosure, nor do they have any other organelles that are with a membrane-bound enclosure. That's very significant. As you will see, the distinction between archaea and bacteria. Bacteria effectively can grow anywhere. Archaea or archaea, which are interesting today, more so, even though they are the oldest, only live in extreme environments. Volcanic caldera, bottom of the ocean, salt plains, polar ice areas, that kind of stuff. But because of those extremes, they have some interesting properties that are that that potentially could have relevance to us down the road. Okay, and so I've never really seen that akaryotic name that they're commenting on here. And so we'll take a look at all these from the so-called common ancestor. When I learned about this, we used more of a table that was like that. Okay, and we're going to do it a little bit differently because what we use in science now is the domains. Okay, when I was taking it, we had kingdoms. You had animal and plant kingdoms, and that's you're going to see particularly that for years they thought that bacteria were really part of the plant kingdom. Why? Because they have a cell wall. So that's where you'll hear the term in the hospital, depending on, yeah, we call them resident or normal microbes, but you'll hear terms like normal flora. Remember, everything was flora, plants, fauna, animals. So we learned about normal flora. Interestingly, I got my doctorate before that domain thing was published. I know. Just shut up. They're all looking at you going, he's freaking old. So eukaryotes, single or multi-celled, obviously can become very complex, et cetera, et cetera. And they're showing you these kinds of things that, and understand that we haven't been around that terribly long compared to the grand scheme of things that are there. And this is part of the evolution. And I'm really not into the evolutionary stuff, but this is important. Microbes are everywhere. And that's really I mean, if you wanted to have one word about the first key issue with regard to microbiology, ubiquity. They're everywhere. Every time you inhale, there are microbes. If we didn't learn that during COVID, we were never going to learn it, even though we all knew about it. Every time you take a breath, there are bacteria, there are viruses. That's the beauty of your immune system. It's designed to limit those. I mean, I don't know what you guys are mostly doing high school stuff when that first lockdown came. So you weren't here like in the fall of 21. Were you? No, you were here in the fall of 22. So you were in high school back then, right? I don't know how they were doing it there. I mean, this classroom would have 10 people in it, and we did a thing called virtual rotation. So we had 10 people. We could have 40 in the class normally. So you only were allowed to have 1 and 4, and everybody had seats that were separated. And little signs, you can't sit here, but you can sit here. And so that's what we did. Week 1, we'd have group A. Week 2, we'd have group B, and we flip flop it. Not a lot of fun. And I'm sitting there with a mask, which when you get older, you can't stand those things. Okay. So they're everywhere, deep in the Earth's crust, all those areas where we see archaea, a big deal within other living things. Okay. And yes, the concept of being essential for life. Without them, we're going nowhere. Let me just give you a heads up on some of that stuff. So let me go back over here to my downloads, and you'll see that in that chapter 1 outline. Okay, so to me, it's a good summary. Vital activities of microorganisms. What do they do? So we can't live it. Nitrogen fixation. When fix does not mean repair in this context. Fix means to affix, to connect. In order for you and I to exist, we have to have amino acids to make proteins and the various kinds of nucleotide bases. Those wonderful, the purines and the pyrimidines you learned about before that are there's the A's, the T's, the G's, and the C's. Those are carbon-based organic molecules that have nitrogen embedded in them. And they're used all over in our bodies that are there. Microorganisms facilitate the attachment of the nitrogen. So that's fixing nitrogen, attaching nitrogen materials, nitrogenated materials to other organic molecules that don't have it. Oxygen. Our oxygen globally comes from algae. Our planet's 5/6 water. The chlorophyll containing organisms in the water are algae, which are a microbe. Okay, that's where oxygen. If you ask me where oxygen comes from here, obviously the trees, the grass, etc., the chlorophyll here in a land-based area, but globally, microorganisms. certainly predominated. So especially aquatic. How do we break down foods? Microorganisms. A great deal of that is done digestively for you. How do we break down waste and sewage? We add microorganisms. Commonly today, to be able to deal with, and it's very interesting, and I talk about it all the time in this course, the pristine nature of our water supply. That's We're lucky. You don't worry about getting a glass of water and being sick. I mean, we have Michigan, there was toxin in the water. Every so often we'll get a boil water directive because there's been a problem. But there's a whole lot of hoops they go through for you and I to have potable water. And depending on where you live, most of us have city water. But even if you have water from a well, then there's a lot of hoops you have to go through to make sure that is. Because if you have well water, you probably have a septic tank, and never the twain shall meet. You will have some significant problems. I have 3 children who experienced problems with that, called giardiasis, when we lived up in the Venango County area for many years. Thankfully, they survived. Okay? So that's just little aspects of it. Food production, my favorite topic. Okay? So how do we make bread? Most of the time, we're using yeast. It's a microorganism. How do we make alcohol? Yeast. Okay? How do we make beer? We use the same kind of yeast organism, Saccharomyces cerevisiae. That's where the Mexican name for beer comes from, cerveza. Just not that you guys would know about that. It's okay. On December 3rd, my granddaughter was 21. God. My eldest granddaughter. And dairy products. Okay? Sour cream, for instance. Just grab one out. Cottage cheese. Cheeses in general are all part of it. Bioremediation. So the oil spills, we have bacteria, many of them have been genetically modified to gobble up oil spills, to work on environmental damage. They almost like have this remarkable capability to surround something and sort of wall it off that are there. Just to give you some idea. All of these things, the enzymes, not just the ones for genetic engineering, but we use enzymes to make cellulose, things when people were all, it's a little bit different. Most of us have in-ear devices, but the lightweight headphones and things like that. Disposable diapers, that came from enzymes that made products that would absorb moisture back in the old days when everybody used cotton diapers and diaper services that were there. Pesticides and antibiotics, high on the list. An antibiotic is a definition. It's an agent that's made by a microorganism that either kills or slows the growth or inhibits the growth cycle of another microorganism. We will learn about this in this first unit in something called the bacterial growth curve. So bacteria, it's survival of the fittest. And one of the ways you survive in a competitive environment is you make stuff to kill the other guy. Just saying. And even better, when they kill the other guy, you know what they do? They gobble it up as a nutrient source. I love that. See, I used to make a joke about one of my favorite cinematic characters, Dr. Lecter from Silence of the Lambs. So if you haven't seen that, go back and see it. I'm having an old bacteria for dinner. The great Sir Anthony Hopkins. A movie, by the way, that was filmed in Pittsburgh. They just sold the house that's out there. They used the old jail downtown for the jail scenes. You can see it all here. There's one that just came out with Christian Bale that was all done in this area. A lot of it in Butler County. It's on Netflix now, whatever their name is, about Edgar Allan Poe. They use whatever. I'll find the name. I'll tell you about it. My wife saw it. I didn't get to see it. I was too busy watching games. So all of those things that are there almost done. Genetic engineering, insulin, growth hormone, interferon, which is made that we'll talk about in viruses, vaccines extensively, A lot of that genetic engineering for the COVID vaccine certainly was part of that. And lastly, gene therapy, which is a little bit different. In gene therapy, everybody, what we try to do is we try to resequence cells. So you take a cell that's doing something wrong and you try to correct it. How do we do that? We have the ability to insert genetic material into a virus, have that virus infect that cell, and effectively reprogram that cell. So we sequence it as RNA, and then we put an enzyme in that causes the RNA to be rewritten inside the cell and the cell's nucleus as DNA. The virus we use for that, and I'll leave you with this, human immunodeficiency virus, HIV virus. So we take the bad stuff out of the HIV virus, resequence it, and use the enzyme called RNA reverse transcriptase. It reverses transcription, takes RNA and makes DNA. That's in its infancy. We're just starting to use that. That's all for today. Exciting. Goodbye. Thank you. Have fun storming the castle.